This is Jose Merino, Editor-in-Chief of the Neurology family of journals. The Neurology podcast provides practical information to neurologists and other clinicians to help them provide better care for their patients. Thanks for listening and have a great week.
Hello, my name's Gordon Smith. I'm a neuromuscular neurologist and chair of the Department of Neurology at Virginia Commonwealth University. And today I have a real pleasure. I get to talk with Tim Miller, who is the David Clayson Professor of Neurology and co-director of the ALS Center at Washington University in St. Louis. Tim is an international expert on ALS and neurodegenerative disease, and he's actually a recent inductee into the National Academy of Medicine. Today, Dr. Miller is joining me to talk about where we stand in development of therapies for amyotrophic lateral sclerosis. We'll spend a lot of time talking about an exciting new therapy called Tofersen and his team's work on achieving a better understanding of disease mechanism leading to therapies. Tim, thank you very much for joining me, and congratulations on the NAM induction. That's super exciting.
Thank you very much, Gordon. I'm delighted to be here.
In May, I think, of 23, I did a podcast with Suma Babu at MGH about Tofersen, and it really rocked my world to hear about this therapy because I will admit I was a little skeptical. We've seen so many disappointing trials in ALS, and of all the podcasts I've done is one that really impacted me. And I'm really excited to hear what's happened over the last couple of years in this therapy.
I think the most we've been able to see, and Suma Babu will probably discuss this with you as well, some of the impressive effects of Tofersen and some of the stories that have come out and some of the individual stories. And what I've been presenting recently and sharing more recently is the 2-year data, where we've looked out at 2 years and just asked some simple questions based on the stories we've heard is like, how many people got better in the setting of ALS? What was the effect that we can see on survival? Some of these simple measures of the success of a therapy. And the data are very impressive. And I think most of the listeners for this podcast will know that most people with ALS succumb to their disease in 3 to 5 years. Some people live much longer than that, but it's a progressive downhill course. And what we don't see in the setting of ALS, though there are some very rare exceptions, but except for those, again, rare cases, we don't see people getting better. And the data that are coming through are not just one person or a few people, that if you look in the clinical studies, 20-25% of the participants in the VALOR study and the open label extension, now looking at it 2 years, are better in terms of function, breathing, and strength. And that's really, to me, a remarkable number.
Tim, I'd like to hear more about the data, but I think most of our listeners are like me. A patient story is something I remember much better than data. Can you give us a story? Like maybe what's the most impressive example you've seen of a patient who's improving after starting Tofersen?
I'll share with you just one story from our center. And this was someone that I was taking care of with ALS. We did genetic testing and found an SOD1 mutation. And at our center, my colleague, Bob Uccelli, was running the clinical study, the VALOR study focused on Tofersen. So I referred this gentleman over to the clinical study, and he didn't do well when he entered the study. He was already not doing well. He was moving rapidly, and his function, his breathing, his strength all declined fairly rapidly when he was in the study. I still don't know whether he was on placebo or getting the drug, but I assume he was on placebo. At 28 weeks, he shifted to the open label extension. So I think, as many know, there was an opportunity in this study to shift open label extension after about 6 months. So he was in this open label extension, and actually I had a Zoom visit with him. And he told me, he said, Dr. Miller, my right arm's getting stronger. This was someone who couldn't use his right arm to lift it above his head. He couldn't really use his arms very functionally at all. He lost a lot of strength. And he said, my right arm's getting stronger. This was circa 2021. I was doing Zoom visits at the time, so it was hard to tell. Around 2021 is when we had announced the first part of this study, the 28-week time point, where we saw really not as much of a difference in terms of function at 28 weeks. We didn't have the opportunity to see the 1-year data, to see the 2-year data as we have now. There weren't as many stories at that time. And so I was skeptical. I thought, wow, really? Is your right arm getting stronger? It's Zoom. You can't tell by Zoom quite as easily. He came in to see us. And when he came in to see us, the changes in his strength were really remarkable. And I think many of the listeners are familiar with the Medical Research Council scale, or the MRC scale, ranges from 0 to 5, with 4 being a wide range of some strength, 5 full strength, and 3 against gravity, and anything less than that, less than gravity. And he had changes going from 3 to 4s and 5s, really impressive increases in his strength, was able to lift his arms above his head, use his right arm as he could show me to pour from a gallon jug. So he was really much stronger than he had been previously after a few months of being in the open label extension. And so for me, that's one of the most remarkable stories. It's one of the people that I saw myself, measured and examined myself, that I was so impressed by, that made it clear to me that Tofersen was having a powerful effect in some individuals.
How did that make you feel? We've been doing this a long time, and I never thought I would hear a story, honestly, like what you described. Describe that moment for our listeners.
I would say that it came slowly and not all at once. There wasn't one moment. We knew early on that we had reduced neurofilament, and my interpretation of those data has been and continues to be a substantial slowing of the neurodegenerative disease process. We had heard some stories, a little bit here, a little bit there. So it wasn't one piece, but seeing this gentleman and seeing other people and hearing these other stories has been really impressive. I say, for me, this teaches us that ALS is a treatable disorder, which I've always believed, but I didn't have proof that we could treat it and make things better. So it's been absolutely incredible to see people with the disease that 10 years ago that they would almost always or always die from, to see people recover some function has been absolutely incredible. Super motivating to find treatments and for the rest of ALS as well, right? So we have a genetic form that we're able to treat, in my opinion, successfully at this point. We need to be able to treat every single person with an SOD1 mutation to really stop their disease and have them, each person get better and have sustained, et cetera. So I think we still have work to do within SOD1. We're not done, but to see some people recover and to see, I think, many people slow down a lot has just been amazing.
Maybe you could talk about prevention. I think the ATLAS study, Mike Benatar's study, looking at treating patients who have the mutation with early decline in neurofilament light chain. Maybe you can describe how you think that's going, how that might change the field.
We still need to understand that conversion from asymptomatic to symptomatic. We need to understand a lot more about people who carry gene mutation and are asymptomatic. Why? They have the same misfolded protein. They have the same abnormality for, in some cases, 40 years, 50 years. Why does that then convert to damage to the motor neurons and to what we understand as ALS? So there's a lot to learn there. Michael Benatar's ALS study and the Prevent ALS, which is part of All ALS, run by Washington University and Columbia, and now being broadly from many sites, I think will teach us about that asymptomatic group and about converting to ALS. And so I think there's a lot to learn about that and understanding that part of the disease. And I think that's going to teach us understanding why do people get ALS in the first place. But you asked about prevention, and specifically in the setting of Tofersen, which is the ATLAS study from Biogen and led by Michael Benatar. The concept is that we can follow a participant's neurofilament level, and that when neurofilament rises, that would be an indication that ALS is about to start, and that you could put then people on drug or placebo in that study and follow them with a goal of being on drug to prevent the occurrence of ALS altogether. We don't have results from that study yet, so we don't know the answer, but I'm super excited about that study for two reasons. I'm super excited about the concept of following and treating people as early as possible. I think it's clear if we get people on at the 10,000 foot view, getting somebody on drugs sooner rather than later, of course, is going to be better. We know that once you've lost motor neurons, Tofersen is not going to make the motor neurons come back. It may help existing motor neurons reinnervate muscle. I think that's true, and that explains improvement. But once the motor neurons are lost, they're lost. So getting people on drug as soon as possible is going to be key. And I think that's happening in the community, or at least many circumstances now that Tofersen's been approved. And the ATLAS study will get people on drug as quickly as possible if and when they become symptomatic in that study. So that'll teach us about that as well. So I'm very enthusiastic overall about the concept of preventing ALS. And I do think that we will be able to do that in many genetic forms of ALS in the future.
What are the implications, do you think, for patients that have sporadic ALS? And maybe you could talk a little bit about your work and how you link the remarkable findings in clinical experience and in the long-term open label experience data with Tofersen to disease mechanism that might be targetable for a broader number of patients.
A couple of things. First is the biomarker neurofilament. In this study, we clearly learned that neurofilament goes down, and in our study it went down substantially by more than 50% with an effective therapy. Will that be true in all therapies? I think we still don't know, but it is an indicator that the biomarkers related to ALS can move with an effective therapy. And many people are using that, and I think we'll continue to use neurofilament as an important readout, as a readout of a lowering of neurofilament being a reduction in the neurodegenerative disease process. That is a substantial move forward. And you can think about clinical trials that are focused on neurofilament, and those are out there now. You might get an early signal. You can argue that you may leave some things behind, but if a drug lowers neurofilament, that's a very promising start. The other, and to me, the most profound learning for non-SOD1 ALS is that ALS is treatable. And again, I've mentioned this already, that believe that for more than 20 years now, but wasn't as clear. And now we see with the right drug and SOD1 lowering in the setting of an SOD1 genetic disease, we can see people slow down substantially and in some cases improve. And to me, that teaches us that if we have the right drug, we can have a substantial impact on ALS. And in some cases, even get to improvement. So I think that's probably, again, the most important learning for me. And I think that we'll continue to learn from the Tofersen story. What are the biomarkers that change? There's many groups that are looking at what are the proteins in the CSF that change? How does healing happen? Are there adjunctive therapies that we can add in? Our group at Washington University, colleague Sean Smith, has started thinking very hard about what should the rehab program be in the setting of ALS. And so people getting better, how do we help them get even better? How do we accelerate healing? And so those are some of the other things that we'll learn about all forms of ALS from these studies.
So I'm curious, the message that ALS is a treatable disease is really powerful. I've heard you talk about this before, and I routinely, when I'm speaking to the community, give them this message. It's incredibly powerful. Have you changed how you talk to patients with sporadic ALS about prognosis or the field based on this experience?
I'm reflecting on that. I changed how I talk about research. I've changed how I view the landscape of drugs that are out there. I've changed my hope for the future in terms of how I discuss this with sporadic ALS patients that I see. But I also need to be careful because in terms of patients that I'm seeing today, there's not a drug like Tofersen that I have for that patient that I'm seeing today. And drug development takes time. So I'm balanced. I want to be cautious with and I don't want to lead people to the impression that I know right now that there's a drug out there that can change the course of their ALS. I do hope that there is. I do think that there's one that's going to be developed in the future that can change the course of ALS broadly, or the sporadic ALS as well. So I'm more hopeful. But I think for the sporadic ALS population that you and I are seeing in clinic this week or next week, we need to be balanced about the discussion.
I have another question about the approach to sporadic ALS when you don't have a specific genetic abnormality to target. Do you think that it's likely that there will be development of therapeutics that target a shared downstream mechanism, assuming that there may be multiple different drivers of neurodegeneration and ALS? Maybe you can talk a little bit about work you're doing in the lab to explore this sort of idea.
I do think it's likely that we can find some shared downstream mechanisms, that we can find some ways to treat ALS broadly. I think there's a number of groups, including ours, focused on TB43, for example, which is a molecule that leaves the nucleus, goes into the cytoplasm, becomes aggregated, but it's probably the loss from the nucleus and that dysfunction that is part of ALS. And we see that as one mechanism. Whether it's enough to target that particular downstream mechanism, I think remains unclear, but it seems to be common among most ALS in terms of finding TB43 dysfunction. So I think people will continue to focus on that, as are we, to develop novel TDP-43 focused therapeutics. And then many groups like ours continue to look for ways to modify the disease, other genetic forms of the disease, which we're focused on. And then we have some basic studies trying to understand, how do some motor neurons resist ALS? Why are not all motor neurons susceptible to the disease? I think many listening to this already know, but it's worth pointing out that the neurons that control eye movements, for example, either succumb very late in the setting of ALS or not at all. People continue to move their eyes, even though those cranial nerves, where they are in the brainstem, is right next door to some of the centers for moving other muscles that are often weak in the setting of ALS, for example, movement of the face. I think that's a ripe area for us to understand the basic mechanisms behind resistance, and then to use those to develop new therapies that can help the rest of the motor neurons. So those are just some ideas, but there are many that are coming through and many that are being tested in clinical trials. And one of the super exciting things about drug development right now are all the tools we have. We're using an RNA-targeted therapeutics. There are other RNA-targeted therapeutics. There's DNA editing probably coming through for the central nervous system. It's now available outside of the central nervous system. Better ways to make small molecules, ways to combine things together. There's just a whole host of ways to manipulate pathways that give us opportunities to try manipulating those pathways in the setting of ALS that we didn't have before.
Tim, thank you very much. What a great way to end our conversation. I would say this has really been an inspiring conversation. Thanks very much for joining me.
And thank you. I also just want to recognize that I am delighted to have the opportunity to share this story. But of course, there are many people who have been involved in this story, many investigators, funding agencies, etc. I also want to recognize the brave volunteers, the participants in this study. Without people who show up for this kind of study, which was challenging in several ways, without people who show up for these kinds of studies, we were not able to find new therapies for ALS. So I want to recognize those brave volunteers.
What a powerful story. I think science matters and collaboration matters. And it's a great study. And I think learning about where the field is going and the work you and other colleagues are doing is really inspiring. So thank you again.
Thank you.
This is Stacey Clardy, your podcast editor. If you've enjoyed the podcast, please take a few moments to subscribe, rate, and review the Neurology podcast through Apple Podcasts, Google Podcasts, Spotify, or wherever you listen. And remember, you can always head to neurology.org/podcast for our full list of past episodes. Or you can also search by keyword in your podcast app for any neurology-specific topics you want to learn about.